And so when it has abundant phosphate, so it will store some for polysaccharide reserve is for energy. Polysaccharide lipid is for energy. Sulfur things generally are stored. Maybe inside the cytoplasm, or maybe outside cytoplasm. In periplasm, that is, it may store a sulfur granule or lipid in the periplasm. So when they need it, they will send proteins, enzymes outside to digest it or to the cell for the usage. And carboxysome. Carboxysome is inside structure. Carbon dioxide fixation, carbon fixation, and gas vacuole. Gas vacuole is it has is a vacuole which is able to be filled with air, and the purpose is in water. We like to go to the top layer to protect itself from being eaten. And so gas vacuole is just like a life jacket. So it can rise to the surface or to sink down to the bottom. So called a gas vacuole. Magnetosome means that it's a magnet. It forms a long chain of magnet. So it may be able to detect the magnetic field. So it knows where the top is or where the downside is, because bacteria do not have an eye. So if it wants to go up, go down, it can also follow the magnetic field, because as we understand, the magnetic line on Earth is pointing down. On the downside. So these are the currently discovered inclusion bodies inside important. Endospore. Endospore is also a very important structure. And this one was discovered in late Ferdinand Kohn. And the discovery is of tremendous value because he discovered that some bacteria, when the environment is bad, it will very dry resistant, so will not be killed by sunlight, will not be killed by heating, and so this is a lot of bacteria, even with boiling, even after boiling. This is because when spores fall into this soup, boiling itself will then the spore germinates into bacteria. So you find a whole world of nutrient for it to grow. A way for scientists to sterilize. Sterilize means to kill all. It's called sterilize or sterilization. Sterilization process is to try to make a certain place, make a certain surface without living bacteria. It's called sterilization. So this currently we know a lot about the sporulation process. First, it is resistant to heat and chemicals, so it's very important. Second, it's is a gram positive cells, is in a few genus of gram positive bacteria, such as Bacillus clostridium, uh, Bacillus form cells, which cause many kinds of disease, such as Bacillus. And Clostridium is also gram-positive. It's a spindle-formed gram-positive cells. So these two genus of gram-positive bacteria form endospores. Some archaea seem to form endospores. So the ability to form endospores is quite limited. Is a return from a spore state to vegetative state. Vegetative is called growing state. Growing state. Okay, let me show you the endospores. This is in Bacillus anthracis. Anthracis is the bacteria forming the anthrax. Anthrax kills animals, and also is one of the most terrifying bioterrorist weapon in the world, because it can be kept. So you can see this is and the process is like this. This is studied by Harvard professor Losick for many years. In much detail. That is when in bad situations without can no longer live in this environment, then it can undergo the final plasma membrane cell wall grows inside to seal off this daughter chromosome. Inside the structure. And this one express many sporulation genes. These sporulation genes will make the coat, make daughter chromosome to shrink into a endospore. So eventually this endospore has the and then eventually this is degraded. That is, this part, mother is dead and daughter is alive. Daughter was released. Is released. So this is the formation process. The germination process, that is if the endospores is heated, not to death, heated to higher temperature, and at the same time, after being heated, it encountered some amino acid, some liquid, some good imbibe to take in water. and then expand and start protein synthesis, and then to break open chip cells. That is a live bacteria. So this is very important for the bacterial life. Okay, we have to introduce you. Let me take it better. Take it. And the first is the outside. Outside, generally called sugar curd is a general name for the outside structures. It's sometimes called tight structure. Tight structure means that if you use water, you cannot wash this structure away. If it's a slime layer, you can use water to wash it away. This is a loose structure. Both are generally polysaccharide. Both are generally polysaccharide. So this structure generally can not swallow it. That is, sugar has to has a hold and handle. So it can escape phagocytosis by the cell. So these are important ones. And then the appendage important for their functions. The first is the large appendage called the flagellum. Flagella is called a whip. It's the Latin name of whip. So it's a large structure. The large structure has a hook and a filament. This hook is connected with 4 wheels to the cell surface as a membrane. So these 4 wheels plus 1 hook is the original structure for the flagella. Then the outer structure is called the exported from here, be assembled outside. So become a long flagella. Forward. If it turns clockwise, then the cell will tumble. Appendage for the bacteria, it's called the flagella. Very important. It's present in both gram-positive and gram-negative cells. Okay, you can see that flagella is a single hair. Peritracheus is around the cell body. It's called peri. It's around with the flagella. Lofo means a tuft on the one end. So these are the 4 different arrangements. It has their function in propelling cells towards a place where they can find a mate. Okay, and this motility is called taxis. If it's moving toward the chemical, it's called chemotaxis. Okay, and so you can see this is the counterclockwise. Then they will be able to move in a straight line, not in a straight line, move towards a definite direction. However, if they turn in that constant direction, it's called a tempo. Then it new direction. So this is that is when they detect desired direction, they will move. But if they detect that this direction is wrong, they will tumble. Then they find another direction to go. So by run and tumble, the cell is very important. Then finally, we have 10 more minutes. The cell wall, emanating from the cell wall, or perhaps from the plasma membrane. That is, the function is to form biofilm. Biofilm is and there's a structure. Its size is between fibrillary and smaller than the flagella. It's called pili. Singular is called pilus, pili. And sometimes it's for conjugation. Sometimes it's a virulence factor. For conjugation means that this pili is and define that bacteria as a female. It defines so this is a very interesting story. That is, bacteria was found to have sex, and this sex is transmitted to the female. And later on, scientists understand that this pilus is bioplasmid, that is a foreign parasite chromosome. The parasite chromosome, in order to contaminate, transmit to another bacteria, so it makes this pilus, and use this pilus to a female. And if there is a the surface with a molecule, so that the pili, when attaching a male, it cannot latch onto the male's body. Only a female is liable to be latched. So the male the female and male together, and then transfer. So this is called a conjugation. Plus. Okay, finally, let me introduce some metabolism. The basic concept of the bacterial metabolism. Okay, metabolism. Metabolism generally divided into two catabolism means degrading the nutrient so that the cells is able to use it for energy. And anabolism is the bacteria can use building blocks to make proteins or make cell walls or to make another bacteria that is build up itself. To degrade nutrient to generate energy. So we know there's a catabolism, right? Using the outside the proteins, polysaccharide, lipid, and degrade into the basic building blocks, and then through glycolysis and also aerobic and to generate ATP to generate energy, or from the TCA intermediate, and try to go into the anabolic process, that is to build up protein or build up lipid or build up DNA. So this is the purpose of catabolism, is to get energy. Anabolism. So these are the important processes: the glycolysis, respiration, and the TCA cycle. So here are the general if the degradation product has no oxygen, then some final product is lactic acid. If in the presence of oxygen, then further oxidation can undergo. And then the waste product is water and the carbon dioxide. So fermentation is degradation. With the help of oxygen. Okay, some the fermentation product may be different. Some may be the alcohol, some may be the alcohol. Some may be the formic acid, some may be the acetic acid. So different bacteria can produce through fermentation different products. So human beings can use these different products. So the most important is alcohol, making wine. Okay, others are lactic acid, lactic fermentation. All sorts of vegetables, and I don't know. They produce different kind of acid, so we can use this kind of different kind of analytic chemicals to identify this acid. So these will help distinguish different kind of bacteria. So, and respiration has two kinds. One is aerobic respiration. That is respiration in the presence of oxygen. That is the hydrogen, the ion, the electron. With hydrogen to produce the water. There is an anaerobic respiration. This is important because without oxygen, still the electrons can be delivered as reduced to nitrous. Nitrite is reduced to nitrogen gas. such as sulfur, elemental sulfur reduced to sulfur hydrogen sulfide. Sulfuric acid is reduced to hydrogen sulfide. So these processes are the field of bacterial physiology, which are specific to the bacteria, not a human being. We human beings has to have oxygen to have the respiration. So these are the differences. Briefly discussed the structure as well as the metabolism of the delay in the original setup. So we have a delay. So I have to also use up some of your time to read the PPT and also find the book to read. The book Brock's Biology of Microorganisms. It's a more detailed, advanced introduction to microbiology. English textbooks, you can also read it. So you can read the chapters and then read the PPT, then you will harvest a lot of knowledge. Okay, thank you for your attention. And we have a second lecture tomorrow afternoon. Until that time, see you later. Thanks very much. Bye-bye.
Bye-bye.